Welcome to Share Food. We start Chapter 1 of Cambridge O-Level Biology 5090. Chapter 1 is Cells. In this video, we will cover the entire point. At the end of this video, students understand this chapter thoroughly. Let's restart this chapter. What is cell? This cell is not a battery cell. In biology, cells are basic building block of life. Cells are simplest unit that have all characteristics of life. Hence, it is a living thing. This is the definition of cell. Take a look at these cells: plant cell, animal cell, and bacterial cell. Structure of plant cell. Plant cell are fixed rectangular shape. Let's relabel the parts of the cell. Cell wall, cell membrane. Cell membrane is also called plasma membrane or surface membrane. Mitochondria, ribosomes, tonoplast, vacuole, starch grain, chloroplast, Golgi vesicle, Golgi apparatus, smooth endoplasmic reticulum, cytoplasm, rough endoplasmic reticulum, nuclear pool, nucleolus, nuclear envelope. Nuclear envelope is also called nuclear membrane, nucleus. Structure of animal cell. Cell membrane. Cell membrane is also called plasma membrane or surface membrane. Nuclear pool, nuclear membrane. Nuclear membrane is also called nuclear envelope. Nucleolus, nucleus. Smooth endoplasmic reticulum, cytoplasm, vacuole, ribosomes, centriole, rough endoplasmic reticulum, lysosome, mitochondria, Golgi apparatus, Golgi vesicle. All these components of cell are called organelles. Structure of bacterial cell. Pilus, pili, capsule, cell wall, cell membrane or plasma membrane, cytoplasm, plasmid, this is a circular DNA, flagellum, the plural of flagellum is flagella, nucleoid, this is a chromosomal DNA, ribosomes. Some bacteria may have flagella and some may not. Features of bacterial cell. Bacteria are often unicellular, means single cell. Bacterial cell wall is made up of peptidoglycan, means made up of protein and carbohydrate. Bacterial cells have no mitochondria and no nucleus. Some bacteria may have one or more flagella. Bacteria are smaller than the plant cell and animal cell. The size of the bacteria is 1000 nanometer. Function of cell wall. Plant cell and bacterial cell have cell wall. Cell wall never exists in animal cell. The cell wall of plant cell is made up of cellulose. Paper, which is made up of cell wall, is also made up of cellulose. Cell wall never controls the exchange of material. The function of cell wall is to protect and support the cell. For example, if cell absorbs a lot of water and becomes turgid, then the cell wall stops it from bursting. This is the function of cell wall. Remember, students should not use common words. Students should use biological terminology, such as turgid. The strength comes from the cellulose, which form fibers having crisscross over one another. Another function of cell wall is to keep cell in shape, form fixed rectangular shape. Function of cytoplasm. It is a clear gel-like consistency. It is nearly all water. It has enzymes to carry metabolic reaction. Hence, cytoplasm is a main site where metabolic reaction take place. Function of organelles. Function of chloroplast. Chloroplast never found in animal cell, but mostly found in green parts of the plant like leaves. Not every plant cell have chloroplast. Example, root has cell. This cell found in plant roots. Red flower and white flower. Chloroplast contain green pigment called chlorophyll. Because of chlorophyll, plants appear green. The function of chlorophyll is to absorb energy from sunlight, and this energy is used to make food for the plant by the process of photosynthesis. Photosynthesis occurs due to chlorophyll. Chloroplast often contains starch grain, which have been made by photosynthesis. Function of vacuole. Plant cells have large central vacuole which contains solution called cell sap. Animal cells have small vacuole which is temporary. In plant cell, the membrane of vacuole is called tonoplast. Vacuole is storage organelle. Function of nucleus. Nucleus contains genetic information. These information are kept on chromosomes, which are inherited from organism parents. The chromosomes are made up of DNA, deoxyribonucleic acid. This DNA has code for synthesizing protein like enzymes and antibodies. Chromosomes are very long and thin, cannot be seen under electron microscope. But when cell is dividing, they become short and thick and can be seen with a light microscope. Nucleus has nucleolus. Nucleolus synthesize ribosomes. The outer membrane of nucleus is continuous with the endoplasmic reticulum. The tiny little dot called ribosomes attached to the network of membrane runs throughout the cytoplasm called rough endoplasmic reticulum, while the one without the ribosomes are called smooth endoplasmic reticulum. Function of mitochondria. Mitochondria are called powerhouse of the cell because it releases energy. Please do not use this word produce energy, as energy cannot be created or destroyed. Mitochondria produce ATP, adenosine triphosphate. ATP is universal energy currency in living organism. The cell that use a lot of energy have a lot of mitochondria, example, muscle cell. Function of ribosomes. Ribosomes are the tiniest structure in the cell. These tiny dots attach to the network of membrane that runs throughout the cytoplasm called rough endoplasmic reticulum. Ribosomes attached to rough endoplasmic reticulum are called fixed ribosome. Those ribosomes are scattered freely in cytoplasm are called free ribosomes. Ribosomes are the site of protein synthesis. Function of centrioles. Centrioles found only in animal cells near the nucleus. The role of centrioles helps in cell division. Function of cell membrane. Cell membrane exists in all cells like plant cell, animal cell, and bacterial cell. Cell membrane lies outside the cell. The cell membrane is a thin layer of protein and fat. Cell membrane said to be partially permeable. Cell membrane controls the exchange of material, control what enters and leaves the cell. The cell membrane is porous in nature. means they have small pores in it. The cell membrane is flexible. The cell membrane is also known as plasma membrane or surface membrane. Common structures between the plant cell and animal cell: nucleus, ribosomes, cytoplasm, cell membrane, mitochondria, Golgi apparatus, rough endoplasmic reticulum, smooth endoplasmic reticulum, lysosome. Difference between the plant cell and animal cell: structures found only in plant cell are cell wall, chloroplast, central vacuole. Plant cells are regular in shape, means they are rectangular in shape. Structure found only in animal cell: centrioles. Animal cells are round or irregular in shape. Relation between cell structure and cell function: cells are specialized to carry out particular functions. The structure of cell is related to its function. For example, nerve cell has function of communication. Muscle cell has the function of contraction. This helps in movement. Red blood cell has function to transport oxygen from lungs to the rest of the body. A root hair cell has the function to absorb water and minerals. Level of organization: cells are smallest and fundamental unit of life. When a group of similar cells that work together is called tissue. When a group of tissues adapted to perform a specific function is called organ. For example, esophagus, stomach, liver, eye. A group of organs that work together to perform a certain function in an organism's body. For example, circulatory system, respiratory system, nervous system. Summary: organelles make up cell. Cells make up tissue. Tissues make up organ. Organs make up organ system. Organ system make up organism. Let's see some of the examples. Examining cell under light microscope. The cell cannot be viewed with the naked eye, so scientists use microscopes to study them. Microscope is an instrument that magnifies an object. Let's examine the plant cell. To examine plant cell, we use onion epidermal, but first we have to prepare the specimen. To prepare the specimen, we need glass slide, cover slip, scalpel, forceps, onion, methylene blue or iodine solution. We use these stains to stain the cell. Staining technique is important to enhance the visualization of the cell. We take a fleshy leaf from an onion bulb, then we peel off its epidermal layer with the help of forceps. We only need small portion of epidermis. Add 2 or 3 drops of distilled water. Add a few drops of stain. The stain we use could be methylene blue or iodine solution. Place the stained specimen on the glass slide and cover it with the cover slip. Before covering, make sure there is no air bubble in the specimen. After covering with a cover slip, if the stain spread across the cover slip, use paper towel to absorb excess stain. Place the specimen on the stage of the light microscope. View specimen under low power magnification, which is 4x. Then view the specimen under high power magnification, which is 10x or 40x. We observe that onion epithelums have rigid boundaries called cell wall. This dark-stained dot is nucleus, which is near the periphery of the cell. This is the cytoplasm. It is impossible to view organelles under a light microscope. These cells are rectangular in shape. They appear like brick of the wall. Under high power magnification, like 10x or 40x, we see a few number of cells are visible, hence the image is magnified. Let's examine animal cell under a light microscope. To examine animal cell, we use liver cell, but first we have to prepare the specimen. To prepare the specimen, we cut a very thin section of liver cell. Place the section on the glass slide. We then stain the cell with the help of methylene blue or iodine solution. Add 2 or 3 drops of stain. Wait for a couple of minutes, then cover it with cover slip. Before covering with the cover slip, make sure there is no air bubble. After covering with the cover slip, if the stain spread across the cover slip, use paper towel to absorb the excess stain. Place the specimen on the stage of the light microscope. View the specimen under low power magnification, then view the specimen under high power magnification. We observe cell membrane, nucleus, and cytoplasm. In paper 6, students may ask to identify these cells. This is onion epithelis, and this is an animal cell, the liver cell. Students should also identify the parts of the cell. Sometimes, students may ask to draw cells shown in any above figure. For example, students may ask to draw fluorescent cells. Let's redraw these fluorescent cells. Students cannot draw their imagination. Students have to draw what exactly they see in the figure. How to use a light microscope. The specimen is viewed in eyepiece, also known as ocular lens. This has a magnification of 10x. Light microscope has 3 objective lenses, which has magnification of 4x. 10x and 40x. The maximum magnification achieved from a light microscope is around 2000 times. To calculate the magnification of a light microscope, simply multiply the magnification of the ocular lens by the magnification of the objective lens used. For example, 10 from the magnification of the ocular lens and objective lens we used is 10, so 10 multiplied by 10 is equal to 100 times magnification. The picture obtained from a light microscope is called photomicrograph. Electron microscope. The cell can be viewed with much detail under electron microscope. Organelles are visible under electron microscope. Electron microscope is more powerful than a light microscope. These are the images obtained from electron microscope, plant cell, and animal cell. We observe cell wall, plasma membrane, chloroplast, vacuole, nucleus, cytoplasm. We observe plasma membrane, mitochondria, rapid to plasmic reticulum, nucleus, Golgi apparatus, cytoplasm. Students should identify these organelles. Such images may appear in exam. It may ask in paper 6. Another picture of animal cell. The largest organelle is a nucleus and then mitochondria. In plant cell, the largest organelle is a nucleus and then chloroplast. The picture obtained from electron microscope is called electron micrograph. Difference between magnification and resolution. Magnification is the ability to bring out a small object seems large, such as microscopic specimen. Resolution is the ability to distinguish two points that are very close together. Resolution is the fairness, sharpness, and clarity of an image. Electron microscope produces a better resolution image, while under a light microscope specimens seem clear but not in details. Calculating magnification. It is important for students to learn how to calculate magnification. First, understand few terminology. Scale bar, a short line usually drawn on a micrograph to show the magnification. This is the scale bar. The actual size of the cell is 1 micrometer. The image size is measured with the help of the ruler, which is 7.6 centimeter. To calculate magnification, use this formula. Convert 7.6 centimeter into micrometer. 1 centimeter is equal to 10,000 micrometer. Before putting the values in the formula, make sure all values have the same unit. Put all values in the formula to calculate magnification. The easiest way to remember the formula, use this triangle. To calculate magnification, you cover M with your finger. You will see image size by actual size. To calculate actual size of the cell, put your hand over A. You will see image size by magnification. The same goes for calculating the size of the image. Let's recalculate the actual size of the bacteria. The magnification of the bacteria is 90,000. We measure the length of the bacteria from the ruler, which is 8 centimeter. Put all the values in the formula. This is the end of the topic. Show love and support by contacting us to make more content for you.